Okay, yeah, welcome everybody. We have a whole string of questions here, so I think we can maybe proceed direct to the questions because we're running a little bit behind time. So the first question, very interesting. It says, is ergo present in psychedelic mushrooms? So maybe I
have a lot of knowledge about that. Actually, I don't have any knowledge about it, but I'm sure it's there, but it's probably not what people are looking for. And, it hasn't been measured as far as I know.
I would guess it probably is, but I would recommend you have regular mushrooms, certainly in Singapore. Now, is it better to take an ergo supplement directly or to get it from natural sources like mushrooms? Again, I'll ask Bob for that.
Well, I think being a food scientist, we try to recommend people get their nutrients from food. And of course, that's my interest is there enough present in the food supply, especially in the United States, the way we tend to grow our food. So if you're not able to get enough, which may be the case, then taking a supplement, I think, is useful. In fact, my wife and I, we both take a supplement every day because we don't rely on our food to provide enough of it, I guess.
Okay, thank you very much. Now, what is the cause of ET's decline with age? Maybe the answer is we don't quite know, but my guess is that many nutrients, the ability to absorb them declines with age. So maybe OCTN1 declines with age. Ergothioneine retention in the body also depends on renal reabsorption. So, and again, kidney function declines with age, so you maybe don't absorb it as well. There are also genetic polymorphisms in the gene that encodes OCTN1, which can affect transport function. And we've seen these in a number of subjects where their ability to absorb ergothioneine in most people is very efficient. In some people, it's a bit slower. Because it came from mushrooms, are there any problems with the people who have allergy to mushrooms? I think it's another one for you, Bob.
Well, I would think anybody has an allergy to mushrooms, they wouldn't want to get their ergothioneine from mushrooms, obviously. So I
think that makes sense.
Yeah. I don't know what else to say.
Okay, based on the several studies, what is the safe dosage to take ergothioneine supplements per day? Maybe I'll ask both of you to comment on that.
For the safe dose, for the U.S., I just mentioned in one of my slides, in the U.S., the two grass approved by FDA, or recognized by FDA, I should say, is 5 milligram. In EU, is 30 milligram for adults, and children 20 milligram. So as you can see from the human clinical trials conducted so far, there's about 4 or 5 Bob, and I just presented is using like 25 or 30 milligram per dose, showing good results.
For me, I don't know. So, yeah.
Yeah. And again, it goes back to this question. We know that there's a minimum level below that. You appear to get problems, increased risk of disease, but we don't quite know what the optimal level is. That's actually true of a lot of nutrients, including vitamin C, E, B12, and so on, that there are deficiency diseases, but a lot of evidence that more than the minimum gives you added benefit. So this is something that needs to be recognized. So how can we standardize the measurement of ET? Which method do you recommend, and how can it be implemented in daily life? I'd just like to go back to something Andreas said. one of the problems that plagues the supplement industry is poor quality control. And in fact, she published a very nice paper recently on NAD supplements, which are very widely used. But a lot of the supplements that people buy off the shelf are not commensurate with what the label states. So high quality control is very, very important. So maybe I'll ask our two colleagues here about the measurement of ET and quality control.
For quality control, first, this material is for human consumption. For quality control, we usually in our industry using food grade. Some more strict one using pharmaceutical grade. What it means, produce this material in a strict pharmaceutical guidance or food guidance, GMP condition, registered with the necessary authority, like each country health authority, and have health authority inspection for pharmaceutical grade material. So that's the way we conduct our manufacture. But for the food industry, as you know, the regulation is widely different. So different countries have different regulations. So it is very hard to monitor because there are so many suppliers of all different type ingredients, nutritional ingredients. It's very difficult to monitor all of them for a consumer. It's not only consumer, even in the industry, it's very difficult to monitor the quality. So you have to go through to the trusted brand to get the higher quality material.
As I said in my presentation, because I found in your paper and those from others, the ergo level can be comparable to each other. I think this is because of targeted measurement. But for our study, we try to cover as many signals as possible. Actually, we have 100 green ingredients that could be added to supplement in our analysis. So to cover all these ingredients in the analysis, we can't trim the analysis. So actually, I compare the level of other ingredient with studies from other cohort using a standard method. It turns out, although we have QC and standards in analysis, but the level cannot be comparable. Actually, it's a sacrifice between accuracy and the coverage, I guess. Thank you.
Okay, two similar questions. Are the beneficial effects the same in frozen mushrooms or cooked mushrooms? So maybe, Bob, that's one for you.
Well, what we found is it's very stable, and so freezing it doesn't bother it. Temperatures up to 400 degrees. We put ergothioneine powder in bread and baked it at 400 degrees, and it survived. Being water soluble, though, if you get any leakage when you're taking stuff out and freezing, thaw it, you get leakage of material. It's going to have ergothioneine is going to be in that solution. Or if you boil them in water and then throw the water out, you're throwing out some of the ergothioneine.
Our own studies confirm that it's very heat stable. As long as the mushrooms have been rapidly frozen. There's usually plenty of ergothioneine. One thing Erwin in my lab and his colleagues did was analyze various mushroom soups. And in general, they didn't contain a huge amount of ergothioneine. But then we found that a number of mushroom soups don't actually contain many mushrooms. If you actually read what's on the label, sometimes mushroom is actually a minor component below all the flavorings and so on. So you have to eat real mushrooms, frozen or cooked or whatever you wish to do. The next question is there any research on NMN, nicotinamide mononucleotide, ergothioneine, and their synergistic effects? This is a very important question because many companies are marketing mixtures of supplements and so on. And the assumption is that if you have 6 compounds that all have some protective effect, putting them together will give you an additive or synergistic effect. There are some studies that suggest this is not true. a scientist called Aubrey de Grey looked at the effects of a number of compounds on lifespan in mice in different combinations, and many things in combination did worse than the individual compounds. But it's a very important question. We're starting a program using human brain organoids to look at possible combinations. The NAD one's an interesting one because one of the papers on ergothioneine and mitochondria suggests that one of the things it does is raise mitochondrial NAD levels, among many other things that ergothioneine can do. So, any comments from anybody on synergism? No? Sorry, go ahead.
Yeah. Actually, it's one of our research interests because, as I mentioned, we have more than 100 ingredient other signals. We try to see whether there's synergetic effect, especially with other supplement in the market, such as Equal. It's going to be hot these years, but will take a lot of computation and time to finish this because we need a clear hypothesis to do this. Otherwise, whole data-driven, and I don't trust this result. Right.
Okay, good. Do all tissues have OCTN1 receptor, the transporter, and can it be absorbed in different ways? And they've raised the question of the mouth epithelium. As far as we know, all organs possess the OCTN1 receptor. The only one that may not is, believe it or not, the human blood-brain barrier. We're not quite sure how ergothioneine gets into the human brain, but it definitely does. So that's one issue. We've actually never tested the mouth epithelium, so many of my lab groupers are in the audience, so maybe we can take a few swabs and look for it. So that would be something very interesting. Another big area of research really is the interaction of ergothioneine with the microbiome in the stomach and the colon. Particularly if you start taking supplements, some of it will pass through into the colon and be a meal for various colonic bacteria. So any other comments from anybody on? Okay, I think let me just check. No, we've got a whole new set of questions that have just come in. I thought, thank God. What is the recommended daily dosage for sleep? And is it to be taken every night? So you've been involved in a number of these studies, so perhaps you can comment.
Based on, because I'm an epidemiologist on diabetes prevention, so we did some studies on sleep patterns. Is there some very interesting result on this? The average sleep duration in America is shorter than the British, which is about 6 hours and then 7 hours, which is not sufficient because at least the curve is like a U curve, and the bottom is around 7.5 hours per day. But for the Chinese guys, we have a habit of napping. Napping is healthy for Chinese, but not for British.
No, I do it during lectures sometimes.
Okay. One of the reason is possibly because the Chinese, I think most Chinese have a habit of napping in the mainland. We also found that the sleep, there's a pattern because some people sleep very late in the weekend, and then on work days they're good. But all these are associated with health consequences. But it's a general comments.
Yeah. The studies with sleep really have not been rigorously done, which is why we've obtained funding to try and do it properly with our sleep unit at the National University. So, but there are papers mainly from Japan that have used various doses, so you can actually look at those.
My comment is it didn't help me last night.
Can you comment on why the study by Zajac et al. Missed the primary outcome, and can we learn anything? This was the study that was presented on what I call the worried well, that is people who think they have cognitive impairment, but in fact when they're tested formally, they don't. But that's a difficult population to use. And the study was also quite short. Our studies with people who do have cognitive impairment run for 18 months or ideally 2 years, because there's a variability in cognitive testing, which is why you should do multiple tests. And also that you need to leave a period of time before you do the tests again, because people can actually learn from the tests. What is the current evidence regarding ergothioneine supplementation for individuals with dementia? We've studied cognitive impairment. I don't know of any study where they've actually used people with frank dementia. I don't know whether any of you.
No.
So that's actually an interesting question. So I think the answer is it has not been studied. So does ergo increase NK cell numbers? I have no idea. I don't know whether anybody else does. And numbers of natural killer cells. Does it affect the immune system? I think there's a study proceeding, supported by Blue California, on the effects of ergothioneine on the immune system. But I don't actually know where it's got to. Do organic mushrooms have higher ergothioneine? And does plant flow from water offer less nutrients? I think they're talking about hydroponic plant growth. So.
Well, we've never really tested organic versus non-organic mushrooms, so I really don't know. As far as hydroponic is concerned, you know, there's no soil there. There's no fungi unless it's added. And I have a big issue with hydroponic vegetables and everything because I think it's going to, it hasn't been tested, but I assume that there's very little, if any, in hydroponically grown tomatoes or cucumbers or whatever it might be.
Okay. Liver toxicity from ergothioneine, somebody's asking about. When we do our studies with elderly subjects, we did full blood counts, liver function, renal panels, and we never saw any toxicity. There have been a number of big studies in China that suggest it's mildly hepatoprotective based on levels of liver enzymes in blood tests. So, but the effect is very small, and if anything, it's a protective effect. I don't know whether you want to comment on it.
Yeah. Actually, we have such data, but we haven't done the analysis. In the Shanghai brain aging study, there's no association between gut function, the liver function, and ET level.
Yeah.
But for younger population, we don't know. We may do this analysis afterwards.
Okay. Is there a difference in ET levels between wild and farmed mushrooms? So.
Well, as you probably saw in the graph, we did porcini mushrooms, which are only grown, or they're only available wild, and it had the highest amount of anything we've tested. So you begin to wonder, we often think that the ergothioneine may be a stress metabolite in mushrooms, because we notice when you grow mushrooms, it increases with the flush when we grow button mushrooms, and each flush is under more stress, water stress, nutrient stress, and everything. So stress growing in the wild may be the key to why the wild mushrooms, in this case with just the porcinis, have more. That's all I know.
Okay, a very technical question. ET activates hydrogen sulfide production and elevates NAD, which I mentioned earlier. These are two pathways. Is there any evidence of redundancy, synergy, or interference? I don't know, but it's a very interesting question. H2S production from ergothioneine in mitochondria certainly happens. I'm very skeptical that that's the explanation of all its biological effects, but it does happen. The paper being referred to is one of two papers that appeared almost back to back in Cell Metabolism. coming up with completely different mechanisms, and I'm surprised the editor let them both through without asking about that. But this, in fact, happens. Okay, organic mushrooms as opposed to ordinary mushrooms. Is there a difference in ergo content?
Not as far as I know. As I say, we've never really tested the difference. But the one thing I failed to mention and that I wanted to mention now is you see that the plants take up ergothioneine, and I don't think they're doing it for us. And so there's an issue of what it's doing to the plant, and there's some research to show that it has some kind of growth regulating properties in plants, increasing the flowering hormone level, and in that case. The other thing I want to mention is you noticed that animals are taking it up from the plants that they eat, and I think there's a whole area of research available out there. What is it doing to the health of the animals? Agricultural animals, there's health aspects are a big thing, but then what about pets? Everybody, is crazy about making sure their pets are healthy. And as far as I know, nothing has been done in regard to that. Maybe you know. But.
There is at least one company in China developing a pet food that contains ergothioneine, plus a lot of other things, but I don't think they've done any clinical testing. There is a lot of interest in pet dogs and cats because they often develop diseases very similar to humans, especially dementia. So there's a lot of interest in this particular area. Can ergo be commercialized internationally, such as in Portugal? And for the company, this is a company question, so do you collaborate with academic institutions for your clinical trials? So perhaps you can.
Wait. Yeah, Portugal is belong to Eurozone, right? So ergothioneine was approved as a novel food many years ago. It should be illegally purchased this product in Portugal.
And crude extract of mushroom, which kind of extraction method is used, and what would be best to preserve the level of ergothioneine?
For the extraction, the best way is using the highest content of mushroom, right? Like Dr. Beelman mentioned, they generally most frequently use the extraction using boiling water out of the mushroom and then filter it and then evaporate. In that way, I think some of the ergothioneine going to be decomposed. So currently using the freeze drying like a supercritical carbon dioxide extraction under lower temperature that preserve the prevent the decomposition of the ergothioneine during the extraction process. I believe that's a better way.
Just a couple of time questions. Basically, can you take too much ergothioneine? Is there a toxicity of higher levels? My guess is very high levels of anything over a long period are not good for you. Ergothioneine is very well absorbed. We've never seen any toxicity in any of our studies, and I think nor has anybody else. But of course, if you start taking very high levels, you'll saturate the body with it, then a lot of it will go into the GI tract and colon, and one of the areas we need more work is the interactions of ergothioneine and other nutrients together with the microbiome, which is really an area that's coming up at the moment. Okay, we have just about 1 minute left, and we've run out of questions online. So is there any urgent question from the audience? Okay, well, thank you very much. And I can call this session to a close. I'd like to thank my colleagues for their excellent contributions. Thanks.